Hello, and welcome to our presentation on selective androgen receptor modulators, or SARMs. In medicine, hormone therapy is frequently considered and used as a means of treatment for a variety of health issues. Androgens are a class of hormones that serve important functions in the body, in both men and women. These include hormonal stabilization during puberty, regulation of muscle, bone, and organ functions, and controlling the expression of primary and secondary sex characteristics. As such, they are currently used to treat a variety of ailments. Androgens, including androstenedione, testosterone, and dihydrotestosterone, or DHT, act as ligands that bind the androgen receptor. The androgen receptor is one of many receptors in the body bound by hormone ligands. Its primary function is as a DNA-binding transcription factor, though it serves several other purposes as well. Androgen receptors can be found in a multitude of tissues in the body, including muscle, brain, bone, fat, and skin. And this wide distribution of them throughout the body is the source of one of the main problems with treatment via introduction of exogenous androgenic hormones. Let's say there's a particular tissue in which you want the androgen to bind the androgen receptor and exert its effect. The issue is, upon introduction of the androgen into the bloodstream, it will bind all androgen receptors in the body in all tissues in which they exist, indiscriminately. Ergo, a response will be elicited in every such tissue in the body. This includes both the desired effect in the desired tissue and undesired effects in the other tissues. The solution? Selective androgen receptor modulators, or SARMs. As their name would suggest, these compounds are able to bind androgen receptors selectively in the body. As a result, they can be tailored to elicit the effects of AR binding only in the desired tissues and be rendered inert in any other tissues, even those with androgen receptors present in them. Yet how is it that SARMs, compounds that must have some degree of affinity for the androgen receptor, are able to achieve this tissue selectivity? Before we answer this key question, let's take a look at the structure and chemistry of the androgen receptor, androgenic hormones, and SARMs.
Now that you have a basic understanding of what SARMs do, let's talk about the basic structure of the main molecules. The wild-type androgen receptor has three main parts to it: the N-terminal domain, the DNA binding domain, and the ligand binding domain. The N-terminus is predominantly hydrophobic and helps to stabilize bound androgens. The DNA binding domain has two zinc fingers that help recognize DNA sequences for the creation of peptides, and is linked to the ligand binding domain via a hinge region that allows for free motion. The ligand binding domain is where binding happens for androgens like DHT, testosterone, and agonists like SARMs. Here is a basic structure of an androgen receptor with a DHT ligand attached to it. As you can see, the DHT molecule is in the center of the complex and is attached via various bonds. These bonds are primarily hydrophobic points of contact, hence why they are in the center of the complex. In addition to these hydrophobic interactions, there are also three hydrogen bonds. The alcohol group in DHT hydrogen bonds to residues 705 and 877, which are asparagine and threonine respectively. The carbonyl group here is hydrogen bonded to residue 752, arginine. Now let's compare DHT to the SARM known as LGD2226. As you can see, LGD2226 is somewhat similar to DHT. The bonding regions for this molecule are also predominantly hydrophobic interactions, and hence why it fits into the ligand binding domain. The main difference in binding between DHT and LGD2226 is that there are only two hydrogen bonds as opposed to three. They are between the carbonyl group and residues 752 and 711, arginine and glutamine respectively. In addition to this, pi interactions also greatly contribute to binding in LGD2226 and are not found in DHT.
In summary, ligand binding to the androgen receptor is driven primarily by hydrophobic interactions, shape specificity, and hydrogen bonding with arginine 752. SARMs designed to these parameters show high AR affinity. For example, the SARM RAD140 has a KD of 7 nanomolar, as compared to KD values of 10 and 29 nanomolar for dihydrotestosterone and testosterone respectively. However, SARMs bind to AR with high affinity in any tissue where there is AR expression. So how is its tissue-selective behavior produced? The first mechanism involves the enzyme 5-alpha reductase, which is expressed mainly in the prostate and associated reproductive organs. 5-alpha reductase reduces testosterone to the more potent dihydrotestosterone. Thus, testosterone is amplified in the prostate, but not in desired target tissues. SARMs, however, retain high AR affinity and activation potential even in muscles and bone. The second mechanism of tissue selectivity arises from the activation of intracellular signaling pathways. In prostate cell cultures, the SARM S22 and dihydrotestosterone activate different cellular phosphokinases. The pathways that S22 activates are not appropriate to produce an androgenic response in the prostate. On the other hand, S22 and dihydrotestosterone activate similar kinases in bone cell cultures. Thus, S22 can elicit the androgenic function of testosterone and dihydrotestosterone in bone cell lines while minimizing prostate stimulation. Research into the precise tissue selectivity of mechanisms of SARMs is ongoing. So far, research and clinical trials have suggested that SARMs hold great promise as drug candidates to produce tissue-selective androgenic responses.